To use Genomicus, first I need to enter the name of a gene. So I'll start with FGF8 and then select a species. These will become my reference gene and reference species. Then I click on Search and I get by default a field view of my gene of interest. In field view, the display is organized in 4 parts. The top panel contains various menus, information on selected objects and navigation buttons. It remains at the same position on the screen no matter where I am in the display. On the left is a phylogenetic tree of FGF8 where only species that have a copy of this gene in their genome are represented. Thick branches represent the path from the root to the query gene. Blue squares represent speciation nodes and red squares represent duplication nodes. My query gene is here in the middle, in the line corresponding to human, which is the reference species I asked for at the beginning. All other copies of the query gene in other species, called orthologs, are aligned vertically in the same green color. By default, 15 genes are shown on either side of the reference gene. All these other genes on either side have a different color, and all their respective orthologs in other species have the same color as their human copy. On the right is the list of species that contain a copy of FGF8, both extant with a small image and ancestral with a red A and in italics. The name of the chromosome or scaffold or syntenic block that is displayed in the middle is indicated next to the species name. For instance, human FGF8 is located on chromosome 10. Ancestral species that predate the species of the query gene are colored in green, all the others are in blue. By default, the reference species is shown at the top of the extant species, just below its own ancestors. This is called a custom tree and it obviously changes depending on which species is the reference. If I want the tree topology to remain exactly the same no matter which species is the reference, I can switch to default tree topology. Exactly the same data is on the display, but it is presented with a species in a different relative order, which remains the same no matter which species is the reference. I'll come back now to the custom tree topology. Genes have an arrow shape that shows their transcriptional orientation. Because the reference gene and all its orthologs are always shown from left to right, the chromosome segment that is on the display sometimes had to be flipped around compared to its conventional orientation. This is indicated by a double-headed arrow under the chromosome block, like for human chromosome 10, for instance. When a genome contains a gene that is not orthologous to any gene in the reference species, this gene does not have a specific color, such as here in orangutan. When a genome segment does not span the width of the display, this means that it physically terminates here. For instance, the FGF8 gene in mouse lemur is at the edge of the scaffold where it is contained. If I want some information on an element of the display, I can click on it and this creates an extra tab here at the top called Details, with extra information on that element and often extra links, for instance here to the Ensemble, UCSC or NCBI websites. Now let's compare the organization of genes around the FGF8 gene between mammals and fish. First, we would like to collapse all species that have been sequenced at low coverage, because they are often not very informative due to the fragmented nature of their genome assemblies. We call these the 2X genomes, and we can remove these by going into the tree menu and selecting Remove 2X Species. Now, some branches end with a small blue circle to show that some data is hidden there. And for the moment, these correspond to all the low coverage species. For instance, here for squirrel. If I'm really interested in squirrel, I can restore its branch by clicking on the blue circle, but the FGF8 gene is indeed on a very small scaffold where there are only 3 genes, so I will keep it hidden. Now I zoom out in the browser window. So now I will remove all branches that are not placental mammals or teleost fish, which covers essentially the frog and the sauria, with birds and lizards. In the case of frog, I simply click on the red cross that appears when I mouse over the node that I want to remove. In the case of birds and lizards, they have a common ancestor called sauria, so in order to remove the whole group, I first collapse the group by clicking on the node itself, then I click on the red cross to hide it. To further clear up the display, I will hide all ancestors for the moment by going into the tree menu and click Hide All Ancestors. Many internal nodes in the tree are not changed on the same line, reflecting the fact that their corresponding species are hidden. What appears here is the result I wanted to see. The FGF8 gene in mammals appear to have 2 copies in teleost fish. I see this because of the duplication node here that is specific to fish, and the 5 sequenced fish species appear twice each here and here. Interestingly, we also see that some genes around FGF8 in mammals are duplicated, while others have been retained in 1 copy only, with either one of the duplicated FGF8 copies in fish. I also see that the vertebrate node has a duplication here, which implies that I should see a human paralog of FGF8 if I follow this branch to the human genome. I see that it is FGF17 to denote that it is a paralog of the query gene FGF8. The FGF17 gene is of the same green color, but surrounded with a white line as opposed to a black line, as for the fish orthologs. Interestingly, a few genes around FGF8 also have paralogs around FGF17, suggesting that the entire region containing an ancestral FGF gene actually duplicated to produce an FGF8 locus on chromosome 10 and an FGF17 locus on chromosome 8. I can zoom in by clicking here to show half the amount of genes on either side. Now I see many smaller symbols in the regions between genes which correspond to conserved non-coding sequences, CNEs for short. The green ones are conserved among mammals, the red ones between mammals and birds, and the blue ones the most stringent between mammals, birds, and fish. The circles represent CNEs located in the intergenic region, where it is displayed, whereas the square-shaped ones are in fact located in one of the introns of the gene immediately to the left of the CNE. Bringing the mouse over a CNE increases its size as well as the size of its orthologous CNEs in other species. If I find that CNEs clutter the display, I can remove them by ticking off the CNE track in the View menu. Now, all this display is seen from the point of view of the human FGF8 gene, but now I would like to see it from the point of view of the zebrafish FGF8 gene. To do this, I can either click on the actual FGF8A gene in zebrafish and then click on the link Switch to this gene as reference. I can also directly click on the species name with the same effect. Now I see very clearly that FGF8A has several orthologs in other fish, as well as several paralogs, due to this duplication node here, while it has many orthologs in mammalian species. One question I'm asking is that although a cluster of green genes here seem to have stayed together at the same orthologous or paralogous loci in all the other fish species, I wonder where are all the neighboring genes in red yellow here and bluish here. To answer this, I need to go to Align View. So in the View menu, I select Align View, and this shows a kind of multiple alignment of the zebrafish locus, which is my reference here, to a number of extant species and ancestral species. The whole display is similar to PhyloView, but one important difference is that the tree on the left is a species tree, not a phylogenetic tree. For instance, there are no duplication nodes in Align View, and species can be represented even if they do not possess an ortholog of the reference gene. In order to be present, however, on the display, a given species must at least have 2 orthologs of any gene of the zebrafish locus linked on the same chromosome or scaffold. For instance, here Medaka has 2 genes on chromosome 1, while these 2 genes are on ultraquantic 66. When genes are boxed, like here, this means they are contiguous as in the zebrafish. When they are not boxed but connected by a thick blue line, it means that they are contiguous in the other genome, like Fugu here, but not in the zebrafish genome. It is equivalent to a gene gap in the alignment. When genes are connected by a thin blue line, it means that the 2 orthologs are on the same chromosome but separated by any number of genes and not necessarily in the same order as in the zebrafish. For instance, these 2 blue genes on Medaka chromosome 15 are separated by about 23 megabases, and this double-headed arrow here indicates that this gene on the Medaka chromosome is in the other orientation compared to this gene. When 2 genes are on the same line but without any connecting lines, it means that the order from left to right is reversed in the other species compared to zebrafish. So Align View is a convenient way of seeing at once how contiguous stretch of genes in one genome, here in zebrafish, are distributed in other genomes. So back to my original question, I can see that the zebrafish FGF8A locus is distributed over 2 chromosomes in stickleback, for instance. Most of the 20 genes are on stickleback group 6, but 2 are on group 9. If I move to the mammals, for instance here, the primate group, I see that FGF8A and yellow neighbors have remained mostly on the same chromosome in human, whereas these bluish ones here are on 3 different chromosomes. If at any time I want to export the data that is on the display, I can do this either as a text file with the name of the genes in the same order as on the display, or as SVG graphic exactly as on the display or without the hidden nodes, just to get a clear picture. These SVG graphics can then be converted to PDF locally.